Welcome to the first annual Symposium on Mitochondrial Health, Healthspan, and Aging. Cyclic Metabolic Switching, Mitochondrial Plasticity, and Brain Health by Mattson.
We all begin to decline when we're in our 30s and 40s, and then it kind of goes down, and then there's another inflection point between 50 and 70 where there's an accelerated performance. For example, task switching, being able to switch between different tasks and keep your attention focused and keep different things in your brain and be able to recall them quickly. Just as with other organ systems, the risk factors for accelerated brain aging are fairly well established. Metabolic morbidity, so obesity, insulin resistance, and diabetes. Hypertension is not good for the brain. That can increase risk for stroke, but it also can cause age-related microvascular problems. Bad cholesterol, triglyceride profiles, and then lifestyle things, sedentary lifestyle, traumatic brain injuries, risk factor for age-related cognitive decline. That's seen with CTE, that football players and others are developing symptoms decades after the injuries, mild injuries sustained during the games. And then conversely, lifestyle factors can mitigate the risk. Okay, so what happens in brain cells during aging is similar for the most part to other organ systems, but in neurons, they're excitable cells. They experience a lot more stress, ionic, metabolic, oxidative stress than many other cell types. So your neurons are more active than your skeletal muscle 24/7. And so if you kind of just make a rough approximation, the activity of neurons in my brain and your brain right now and the circuits that are processing the information are kind of like doing moderate exercise of your muscles. And so neurons have to have the ability to deal with these stresses. And as I'll tell you, that not all neurons are equal in terms of the stress, the amount of stress normal stress, activity-dependent stress they encounter during their life. And that provides clues as to what goes wrong with neural network activity and aging and neurodegenerative disorders like Alzheimer's and Parkinson's. Okay, so this is just kind of a wheel thing taken from previous work by Judy Campisi and others in the field who had a Cell paper a long time ago. But mitochondrial dysfunction, oxidative damage, impaired autophagy, so ability to remove impaired dysfunctional mitochondria, proteins, aggregates, etc. Impaired repair mechanisms, DNA repair is impaired during aging. It turns out that the progeria syndromes, genetic progeria syndromes, Cockayne and others, Bloom, are caused by mutations in DNA repair enzymes. So that's very critical. And most of that DNA damage that needs to be repaired is caused by free radicals. In the brain, there's what I'll call aberrant neural network activity during aging that is greatly exacerbated in Alzheimer's, Parkinson's disease, and even plays in epilepsy, where it's obvious. And even stroke or traumatic brain injury, it turns out that the inability of neurons to maintain their excitation and maintain their membrane potential within normal limits is compromised. There's this process called excitotoxicity in which, under adverse conditions, whether it's subtle changes that occur during aging or a acute condition like epileptic seizure or a stroke, when the neurons are metabolically compromised, they're very vulnerable to degenerating. So at the middle of this wheel, I put dysregulated energy metabolism, and that has a lot to do with mitochondria, but it could also have to do with things like the systems used to move glucose or ketones or other nutrients, amino acids possibly, into cells. So the transporters for these nutrients. For example, insulin resistance, there's a problem in cells, and it appears this happens in neurons too, in their ability to take up glucose in response to insulin. So that's not a problem with the mitochondria necessarily. It's a problem with the ability to take up the nutrients. We've done a lot of work on calcium regulation. Calcium is the most important intracellular signal that is important for all behaviors, for learning and memory. It's important for regulating growth of neurons, formation of new synapses. It's a major regulator of gene expression, all sorts of different genes, including genes involved in DNA repair, autophagy, resistance to oxidative and metabolic stress, are upregulated by the stress of the neuron being active. So just like when you're exercising your muscle, stress, sodium influx, calcium influx, free radical production. But it's okay because it's a good stress, and our cells have evolved really elegant ways to respond adaptively to the stress. And that's kind of what I'm focusing on today, these adaptive cellular stress response systems. Okay, so quickly, kind of a general idea is that aging leads to those changes I showed on the wheel. And then in combination with environmental factors, genetic factors, you end up getting impaired stress responses, reduced neurotrophic support, protein aggregation in disease states like amyloid and tau and Alzheimer's, TDP-43 and ALS, alpha-synuclein, Parkinson's. And then these age-related changes exacerbate the accumulation of these proteins. On the other hand, these proteins can impair some of these systems. For example, we showed in the early 1990s that aggregating A-beta, when it aggregates in association with the membrane of neurons, it induces membrane lipid peroxidation. That impairs the function of the sodium pump and calcium pump, these ATP-dependent pumps that move the ions back out to restore the membrane potential, and that makes neurons vulnerable to excitotoxicity. So these proteins tend to destabilize neural network activity in a bad way. Okay, so how are nutrients moved into the cells? I'll just talk about glucose and ketones, lactic acid too. So there's glucose transporters in the blood-brain and the epivascular endothelial cells. Glucose moves in. Neurons have a glucose transporter called GLUT3 that moves glucose in. Ketones are moved through the MCTs, MCT1 here in the endothelial cells and MCT2 in neurons. The main circuitry in the brain, and I wrote a book about this after I retired from the NIH. It's about glutamate, the brain's most important neurotransmitter. About 90% of the neurons in the brain use glutamate as their neurotransmitter. They're glutamatergic. It's an excitatory neurotransmitter. So the core circuitry throughout the brain is glutamatergic neurons. Then throughout the brain, there are about maybe 15, 18% of the neurons are inhibitory GABAergic neurons. The inhibitory GABAergic neurons, they just act locally to, and they're activated by glutamate to turn off a signal. So we have some signal coming in, it's important to turn it off. The GABAergic neurons do that. All the other neurotransmitters, serotonin, dopamine, acetylcholine, norepinephrine, those neurons are few in number, and they're located mostly outside the cerebral cortex. The only way they affect our behaviors and function of our brain is by modifying the ongoing activity in these glutamatergic neurons. So a lot of people don't know that aren't neuroscientists. Then I mentioned there's differences in neurons in the brain. It turns out these GABAergic neurons are very active. They're loaded with mitochondria. They fire like crazy. And we think they're the most vulnerable to aging and neurodegenerative disorders because of their high metabolic load, their high levels of oxidative stress. So Andy, I think Andy talked a little bit mentioned differences in mitochondria in different cells. Well, here's a good example. This was Tom Delgado and Pam Yao in my lab. They essentially looked at mitochondria by 3D electron microscope reconstruction. And the bottom line is what they found is that in the dendrites of the neurons, the region of the neuron that receives signals from axons, the mitochondria are very long. So this is pink is a mitochondria. Then in the presynaptic terminals, this is a presynaptic terminal here, for example. It's like this one is loaded with neurotransmitter vesicles. They have short, stubby mitochondria in the axons. And then holds up. So here's just examples of dendritic mitochondria and axonal mitochondria. And we're not sure, if and what these morphological differences in mitochondria relate to in terms of the function of the mitochondria. It's pretty interesting. For example, I have a hypothesis that in the dendrites, these mitochondria are, by signaling mechanisms such as Andy talked about, for example, are coordinating or monitoring activity between multiple synaptic inputs along the length of the dendrites, right? Because this part of the mitochondria you're getting, if this presynaptic neuron was activated, you get the calcium influx here, and you may have local effects here in this part of the mitochondria that then may propagate signals. So I'm retired now. Someone else can look at that. That's an interesting problem. Exercise. I'm going to spend not much time on exercise. I'm going to just say it's good for the brain. There's quite a bit known. Regular exercise in mice or rats will increase number of synapses between neurons. For example, in the hippocampus, will actually increase the size of the hippocampus, the brain region critical for learning and memory. Aerobic exercise increases angiogenesis, the growth of blood vessels. So just like when you exercise your muscles, you get this proliferation of the blood vessels so that they can provide more blood and energy to the cells. We think that's true in the brain. And then there's also signals from the periphery. There's evidence that proteins produced by muscle cells during exercise can go to the brain and do good things in the brain. Some evidence of anti-inflammatory molecules from adipocytes, probably from maybe these are produced higher from healthy adipocytes, I guess. Turns out that exercise can actually cause a kind of a browning of the fat cells. Okay, so then here's an example Alexis Stranahan did when she was a graduate student in my lab quite a while ago. She took normal mice, wild-type mice, and diabetic mice with a leptin receptor mutation. So these mice overeat and they become obese and diabetic. She divided them into 3 groups: ad libitum feeding, sedentary. So this is the normal housing conditions for mice or rats. Ad libitum running wheels and cages, daily calorie restriction with time-restricted eating, sedentary, and then the combination of the calorie restriction and the running. And she let them go for 3 months. She took out 1 hippocampus and counted synapses. In both the wild type and the diabetic mice, the exercise and the caloric restriction with daily fasting increased the number of synapses, and the combination causes a significant, I'd say, an additive effect. And then the other hippocampus, she measured BDNF levels, this very important growth factor that's important for synapse formation, maintenance, learning and memory. And it's also very potent in protecting neurons against oxidative, metabolic, and excitotoxic stress. Ewo Cheng in the lab, now quite a while ago, 13 years ago, published a paper where she found that BDNF, this neurotrophic factor, induces PGC1 alpha, and she looked at that using a luciferase reporter assay for PGC1 alpha activity. And then she also looked at signaling pathways, other signaling pathways actually known to be activated by BDNF, including CREB, and found they were upregulated. And then she used RNA interference to knock down PGC1 alpha first in cultured neurons. So this is an example looking at mitochondria with a fluorescent probe. This is a rat embryonic hippocampal neuron, 3 days in culture. And this is a neuron 10 days in culture. So over this 1 week period, there's dramatic proliferation in the number of mitochondria as the dendrites and axon of these neurons grow. And then they seem to be distributed throughout those processes, which makes sense. That makes sense. You have to have ATP available and the other functions of mitochondria out in these areas, which are subjected to a lot of stress. And the bottom line is she knocks down PGC1 alpha. It inhibits the formation of synapses in these developing neurons, and it blocks the ability of BDNF to increase the formation of synapses. She also looked at this in vivo by using a viral vector with a small interfering RNA to knock down PGC1 alpha, and found that she knocks down PGC1 alpha in hippocampal neurons over a period of several weeks. There's a reduction in number of synapses. So that suggests that PGC1 alpha and perhaps mitochondrial biogenesis are necessary for the maintenance of synapses. So I'm going to quickly go through this. Then I'll talk about mitochondrial sirtuin 3. It's a protein deacetylase localized to the mitochondria. It doesn't seem to be in other cellular compartments, at least in neurons that we've looked at, but other people are finding that in other cells. And so we have SIRT3 knockout mice, and we find that they have hyperacetylation of many proteins in the cerebral cortex and hippocampus. It's shown using a anti-acetylated protein antibody. So this is whole cell, and this is isolated mitochondrial fraction. And then Ewo found that if you put running wheels in the cages of wild-type mice, it increases, well, does a lot of good things, but it will increase, it will decrease acetylation of a number of proteins, including superoxide dismutase 2, which is a very important mitochondrial antioxidant enzyme. And however, in the SIRT3 knockout mice, there's a hyperacetylation, and running is not able to alleviate that hyperacetylation, suggesting a major role for SIRT3 in running-induced deacetylation of mitochondrial proteins. We found that if we, now this just shows SIRT3 increases, running increases SIRT3 protein levels, both in vivo and then glutamate at low levels. If we stimulate neurons at physiological levels, it increases SIRT3 levels. If we knock down SIRT3, or take neurons from these SIRT3 knockout mice, they're more vulnerable to excitotoxicity induced by glutamate, canic acid, or NMDA. On the other hand, if we overexpress SIRT3, their fewer neurons are killed by these excitotoxic insults. And in vivo, the SIRT3 knockout mice neurons in the hippocampus are more vulnerable to excitotoxicity. Another lab showed a little more recently that mitochondrial membrane depolarization will upregulate SIRT3, which is interesting because when neurons are active, the calcium influx into the mitochondria will depolarize the mitochondria. So I think it's an adaptive stress response at the level of the mitochondria. I'm going to briefly talk about intermittent fasting. Most of you have probably heard of this. We found beginning in the 1990s that eating patterns where there's periods of time sufficient to cause a metabolic switch from glucose to fats and ketones over periods of many days and weeks and months do good things to the brain, including protect it against a number of insults that are relevant to Alzheimer's, Parkinson's, and stroke. So anyway, with fasting and also with extended exercise without taking in any carbohydrates or glucose, you get glycogen depletion from the liver and lipolysis in the fat cells. Free fatty acids are released into the blood. They're transported into the liver and converted to two ketones, beta-hydroxybutyrate and acetoacetate, so BHB or AA, and these are actively transported through the MCTs into the brain and into neurons. We found, and I'll show data briefly, that, of course, it's well known that the ketones can be used to produce ATP, and this was actually per molecule of the ketone, there's less free radical production per ATP produced compared to glucose. But others, Eric Verdin was one of the first to provide evidence for this. There's signaling functions of BHB, this ketone. We found it upregulates BDNF. I'm going to skip this. If you're interested in the science of intermittent fasting, there's a lot of places to find it on PubMed. After I retired, I wrote a book on that, focusing on the science. And I should point out, not all of effects of intermittent fasting can be accounted for by a reduction in calorie intake. So we think that the metabolic switching in the fasting component is triggering stress responses that aren't necessarily seen with caloric restriction. There's some early data showing in models of epilepsy, stroke, Parkinson's, and Huntington's disease pre-treatment. So preconditioning with intermittent fasting is neuroprotective. In the stroke model, there's some evidence from other labs that intermittent fasting can enhance recovery, and there's actually clinical trials going on now. Okay, I don't want to run too much over. And so what about humans? Well, before I retired, Dimitros Kapagianis, who's a neurologist who's in laboratory neurosciences under me then as an independent investigator, he and I designed a human study to look at the effects of intermittent fasting on the brains of individuals at risk for cognitive impairment and Alzheimer's disease because of their age and metabolic status. So these are individuals, men and women, between the ages of 55 and 70, and they have obesity, so BMI greater than 30. They also have insulin resistance, but not diabetes. So they have pre-diabetes. And based on epidemiological evidence and other evidence, those individuals are increased risk for cognitive impairment and also caused by Alzheimer's, but also vascular cognitive impairment, which is a different type of dementia. Obviously, they're a bigger risk for stroke. Okay, so these individuals were randomly assigned to one of two dietary interventions. 5-2 intermittent fasting, where 2 consecutive days a week, they ate only 480 calories in the form of a shake. The other group, the USDA Healthy Living Diet group, they were educated on healthy diets and control of calorie intake and calorie reduction. But they they ate at home. So this wasn't, we don't know exactly what they ate. But I'll show you they lost weight. In fact, both groups lost about the same amount of weight. So there seemed to be good compliance over the 2-month period. So what we did then is at baseline, that is before they started these interventions, and 2 months later, Dimitrios and his group did a battery of cognitive testing. They did structural and functional magnetic resonance imaging of the brain, did a lot of blood work. They got cerebrospinal fluid, which we didn't publish in this paper yet, I don't think. And but this is an initial paper that came out last year. I'll show you some of the results. So let's look at kind of some of the basic things. So significant decreases in body weight over the 8-week period. So the red is the intermittent fasting, 5-2 intermittent fasting. The blue is the USDA healthy living diet. So BMI went down, waist circumference went down, fasting insulin went down a little bit more in the intermittent fasting group. HOMA IR, that's a measure of insulin resistance. That went down, which is a good thing in both groups. Maybe a little more in the intermittent fasting, and glycated hemoglobin went down. The ketones, BHB and acetoacetate, were up. They were up more. And this was taken in the morning. So it's after a fast, and it was not on the second day of the 480 calorie diet. So there the ketones were even much higher. This is just going up into the 150, no, it's about 200 micromolar level. So this is just small elevation, but it turns out there's kind of two waves of increase in ketones following the onset of fasting. One that occurs beginning about 10 to 12 hours, which makes sense. These were in the morning. And then the other, there's kind of a delay, and then after 24 hours, and certainly by 48, you get a bigger increase up into the millimolar range. We think this initial 100 micromolar range is actually important for some of the signaling functions of ketones. So what about cognitive testing? The subjects on the intermittent fasting showed significant improvements in executive function, verbal fluency, and set shifting, which is a kind of a measure of task switching. And these were significantly more than the control group. And in fact, the control group, there were no significant change from baseline. So whereas the intermittent fasting is improving memory, the healthy living diet, even with loss of body weight, wasn't. A while ago, almost 10 years ago, Christina Monrosi in my lab, and then Slyman et al. In Moses Chow's lab at NYU showed that the ketone BHB induces BDNF gene expression. So you can look at the mRNA or the mRNA or the protein level. And in the culture, this was in cultured neurons. Interestingly, if we incubate the neurons in high glucose, so this is 10 millimolar glucose, which is like diabetic levels compared to blood, but this is actually what most people put in their cultures. But if we reduce the glucose to 1 millimolar, then we see induction of BDNF, but not in a diabetic level of glucose. And then Moses Chow's group showed that in vivo, if they infuse the ketone into the brain, it increases BDNF. And there's a number of signaling pathways for this ketone BHB that have been talked about. Now I'm going to, in the last, so 20, what am I? Yeah, I'm getting close here. In the last 5 minutes, I want to talk a little bit more about the ketones. So Steve Cunan up in Vancouver, he's done some beautiful PET imaging looking at the uptake of either radiolabeled glucose or radiolabeled acetoacetate into cells in living humans. And what he found in this study, what he did is he had the subjects on a normal carbohydrate-containing diet, and then he switched them to a purely ketogenic diet. And what he found was that when there's, the bottom line is when there's high normal levels of glucose in the blood, the neurons and cells in the brain are mainly using glucose. However, when glucose levels are low, and I'm not talking about hypoglycemic, just low normal, then neurons are switching to using a lot of ketones. And he showed a perfect relationship between blood ketones and the cerebral metabolic rate for ketones. We, quite a while ago, in 2013, published this paper where in one of our mouse models of Alzheimer's disease, we supplemented the diet of the mice with a ketone ester, a beta-hydroxybutyrate ester. So at that time, you could not buy this BHB ester, but now there's several companies that put this in drinks. And as with a lot of good things that you put in the mouth, if you put this ketone in your mouth, it has a very bitter taste. You wouldn't want to eat it plain, but you can put some good tasting stuff in these drinks, but not fructose. And don't put fructose in there. They don't. So anyway, you can buy these things. There's a lot of work going on this now. Bottom line is we found that long-term administration of this ketone ester in the diet reduces the amyloid accumulation, tau pathology, and ameliorates learning and memory deficits. And there's multiple clinical trials of that ketone ester in people with older people with mild cognitive impairment or Alzheimer's disease, including a study headed by Dimitrios Kapagiannis, who did the intermittent fasting study. Now, I'll finish this by saying that we know a lot about what's happening during the fasting or exercise period and during the recovery period. And one of the things that's happening is switching between activation of stress response signaling, conserve resources, recycle things in the challenge period. So upregulating autophagy, for example, upregulating antioxidant enzymes repair. And then and mTOR is down, so protein synthesis is down. And then during the recovery period, mTOR goes up, amino acids come into the cell, and PGC1 alpha is activated. You get mitochondrial biogenesis in this switching back and forth, whether it's daily exercise or twice weekly fasting or whatever. Over time, this challenge recovery scenario seems to be good for cells in many ways. Finally, I'm going to skip that as just showing intermittent fasting also upregulates SIRT3, which seems to play a role in beneficial effects of intermittent fasting on the brain. This last slide is just something for the discussion session because you can impose metabolic stress, mitochondrial stress on cells at a mild level pharmacologically. 2-deoxyglucose, essentially you're restricting glucose availability to be used at the cellular level. Dinitrophenol, it's a mitochondrial uncoupler. At low levels, it's not toxic, and we and others are finding it can be neuroprotective, even with long-term administration. Inhibiting mTOR. And then the other thing we can talk about in the discussion is this idea of transient exposure to such pharmacological interventions rather than continuous exposure, because conventional drugs, the idea is you want a constant level, kind of constantly inhibit some receptor or whatever. With these good stress-based interventions, I think you want them intermittently. So thanks a lot. Here's my group a long time ago at NIH, and this is faculty at Hopkins Neuroscience, and this is actually they're post-bac fellows that we had a running group at NIH. So thanks a lot.
Thank you, Dr. Mattson. Do you have time for one or two questions?
Yeah, if you do, I have time.
Yeah, hi. Thank you for a wonderful talk. I especially enjoyed the part about the ketones. I'm Gino Cortopassi, UC Davis, and we've done about 5 years of study or more on ketogenic diet for extending lifespan and health span. And what we see is that our main hypothesis is that it's reducing inflammation. We see that dose dependently in mice on cytokines. So the more keto you are, the lower the cytokines go systemically. Second, in vitro with A-beta inflamed microglia, there's dose-dependent anti-inflammatory effect of BHB as well. And then in a recently published paper, we see that dose-dependent inflammation that's blocked by BHB can be itself blocked by blocking MCT. So it requires entry of the BHB into the cell. MCT inhibitors, sorry. And then the last one is that in the PS1 APP mouse model, one can rescue memory as measured by long-term potentiation, LTP, with the keto diet, and also by washing the hippocampi with 3 millimolar BHB, and you can get the LTP back by that. And we believe that's an anti-inflammatory process, as you've shown in your studies with BHB. And then the last one is that there's one that just is being accepted now, that it's also, keto is helping in the ApoE4 mouse model as well, and rescuing LTP there.
Yes. So, okay. So your question.
My question is, I don't know if you absolutely need the stress if you just switch the metabolic substrate to a mitochondrial metabolic substrate. So BHB, as you know, is uniquely metabolized in the mitochondria. Maybe you don't need another stress. Maybe you just need to switch to metabolic support.
One way to look at this is that BHB is a stress signal. It's acting as a hormone to alert the brain, for example, to the fact that, hey, I haven't got food for a while. I better figure out how to get food. And
yeah, I agree with that. So it's a stress signal. Yeah, you're saying it's a fasting signal.
Yeah. But, okay, so ketogenic diet. I have mixed feelings about ketogenic diets per se. That the fact that they're ketogenic is good as long as there's not a lot of saturated fats, bad fats, and somehow individuals get the good chemicals that are in fruits and vegetables, which are actually, I think, inducing adaptive stress responses. So, yeah, I don't do ketogenic diet. I'm more plant-based, but I do I essentially fast 18 hours every day, and then I exercise at the end of the fasting period. So, yeah, so I think that the lifespan, I don't know, we don't know from humans, as you know, for most ketogenic diets, if there's saturated fat in rodents, that actually they become obese and don't live as long. So, yeah.
But if it's isocaloric, if you switch the mix to more lipid, as long as it's isocaloric, between the 3 different diets, low carb, or carbohydrate rich, or ketogenic, they don't get fat. So,
yeah, that makes sense. Yeah. Okay, right. But yeah, I agree. The ketones are good, and I wouldn't want people to be lazy though and not exercise or fast because they think that ketogenic diet is just as good.
Alyssa?
Thank you. Thank you, Dr. Mattson, for an amazing program of work. You've always been way ahead of us in this, looking at hormesis and all these pathways. This is Alyssa Epple from UCSF, and I'm going to talk at the end about more clinical studies and short-term studies of how we can induce hormetic stress. And my question for you is translational and about how to apply some of the dynamics of hormetic stress to humans. So the answer might be speculative, but I welcome that. You've thought more about this than most people. We have beautiful models of hormetic stressors in model organisms where we know the dose frequency, and we also know that it's tricky business because when you induce acute stressor, the stronger the better for inducing a hormetic response to a certain point. Then there's an inflection point when it turns toxic, when there's too much oxidative stress and it's more damaging and the recovery doesn't catch up to the stress. And so do you have any principles that you could derive across from what you've seen in humans? So for example, exercise, we know there are high-intensity interval training that's probably been studied the most in terms of the kinetics and the dose response, but what about other stressors, hot, cold, hypoxic stress, from what is a principle to go by? So people use intuition and how they feel in their body, and I don't think that's good enough.
And I think that the data are really strong for exercise, right? And the thing with exercise and the short-term fasting and intellectual challenges for that matter is these are things that we evolved with way back when we were worms, maybe, or maybe even before worms. So whereas, and then, obviously heat and cold, but usually it's not like rapid changes in temperature. I guess it depends on the environment. So I'd say with exercise, intermittent fasting, and the data is strong. When you get to like some pharmacological thing or even some naturally occurring chemical, there's just not enough data to say what's the best thing for us to do. And that's why I'm but I think that work needs to continue. I don't think there's going to be a silver bullet. For example, rapamycin, I wouldn't take it myself. And another thing, like, big problem with the animal studies, rapamycin is a good example because the mice, so in the big three center study with Miller and the NI group and others, and I know we need to get to the next talk, so I'll try to make this quick. The animals are raised in a germ-free environment, right? And you're giving them something that suppresses the immune system, maybe somewhat even at low levels. And so it extends their lifespan a little bit, 5-10%, and it was significant. I don't know. I would definitely not think that's the basis for humans to take it. I know some people are, but I think it's not very wise. We can talk more in the general discussion tonight.
Thank you so much. Great answer.
Yeah.
I have a very quick question. Ketogenic diet versus exogenous ketones, not in the dish, but in the person. How much of the effect that you see is due to the ketones and the effects on sirtuins and BDNF versus the reduction in insulin which is preventing diversion away from mitochondria and ATP production toward other storage factors. In other words, I'm looking. I look at the data on exogenous ketones all the time, and I personally, in humans, don't see it. And there are a lot of companies making a whole lot of money on it. I'm trying to figure out, is it the ketogenic diet because it does both, or the exogenous ketones have some hidden effect I'm not seeing?
I wouldn't say that ketones are the total effect of the ketogenic diet. They're often relatively low-calorie diets. They improve insulin sensitivity, so they actually improve glucose metabolism. We'll see with the ketone ester what happens. There's a lot of clinical trials going on. But the exercise thing hasn't really panned out like the initial findings, but we'll see what happens. There's still a lot of interest from the exercise physiology folks on doing studies with this, manipulating ketones and glucose in relation. For example, there's some pretty good evidence that the ketone ester supplementation may enhance recovery. It doesn't seem necessarily to enhance performance, but there's at least several studies that have found enhancement of recovery. So maybe it's enhancing the second phase of the challenge, recover, repeat scenario cycle.
Well, thank you again, Dr. Mattson. I'm delighted you're going to join us.
Stay tuned for the second annual Symposium on Mitochondrial Health, Health Span, and Aging.